All right, I suppose we can get started. As you probably heard, Kelly and Aaron are both at a conference right now in sunny San Diego, so they're telling the world about their research, which is why I'm here as your substitute teacher. Um, my name is Emily. I'm a PhD student in the same lab as Kelly and as Hao and Aaron. Um, and I actually have a long history with this course myself. I took it as an undergrad, so I've been where you are. I've also TA'd it several times, and I've been the instructor for it, so I've seen it from all vantage points. It's still one of my favorite courses. And it's always been in the same room, too, so I've gotten to know this room very well. It's got the killer sound system, so what can you do? Also, fun fact, I do fMRI research here at the CMRR, at the Center for Magnetic Resonance Research. So I like to talk about neuroimaging and brains, so I'm excited to give this talk today. Any questions before we begin? All right. So let's start with some basics. And I apologize if some of this is review. Bear with me. So we have 4 major lobes in the cortex. The cortex being this outer layer of the brain with all the wrinkles and the folds in it. And you see all these kind of valleys, the darker parts, and these ridges here. These are known as the sulci and the gyri, or a single sulcus, single gyrus. So sulcus, think of as the valleys, and gyrus as the ridges. And all of these lobes are intricate and complex, and they're involved in a lot of different types of processing that we're just beginning to understand. But at the very broad level, we can say a few things about their functionality. The frontal lobe, for example, is involved in higher level processing, executive function, paying attention to things, making decisions, controlling motor movements. And that's this red bit here. The parietal lobe up here in yellow is more involved in somatosensory, so informing us about our external environment through touch and pain, etc. And then we have the occipital lobe, this back part in blue here, and that's largely involved in visual processing. And last, but certainly not least, we have the temporal lobe, this green bit here, and that's the best lobe, at least you'll come to believe that after this class, because it's involved in auditory processing, speech, music, frequency processing, all manner of auditory processing happens there. And definitely put a star next to that. It's possible this will become an exam question. What lobe is the auditory cortex in? Answer, temporal lobe. Boom. You're already doing well. Oh, interestingly, okay, so one way to remember where the temporal lobe is that it's the lobe closest to the ear. So that's cool. But for some reason, the occipital lobe is the lobe furthest away from the eyes. So I'm not sure why that is, but so be it. So there are different ways of dividing up brain structures. We can look at the cellular composition of each brain region. The fancy term for this is cytoarchitecture. So we're basically looking at the tissue of the brain through a microscope, and we're looking at the differences in the cell structure and deeming them as being different parts of the brain because they have different structures. And another term you may come across is histochemistry, which is looking at the chemical makeup of the tissue and the cells using stains and various indicators. And again, you're looking through a microscope. So different stains can behave differently, different indicators for different tissue types. So they'll look different under the microscope, and then you can say, okay, this tissue type is different from this one. And another fancy term is myeloarchitectonics. So you can just use that in your everyday life and sound really smart. But it's actually something you're already familiar with. So you've heard of the terms gray matter and white matter, and the difference between them is the degree of myelination. So basically, myelin is these kind of fatty sheaths that go around our axons or nerve fibers, and they aid in the conduction or movement of electrical information from one part of the brain to the other. So when these axons are myelinated, it improves the speed at which this can happen. So you can think of white matter as being good with kind of global communication. Like, I need to go from the back of the brain to the front really fast, so I'm going to go through these myelinated tracks. Whereas the gray matter is more associated with local networks, local processing. It's largely unmyelinated axons. So because it doesn't have to travel as far as fast, it doesn't have this myelination, and that also means it takes up less space than the white matter, so I guess that's a good thing. And you can see in this picture here, the lighter gray would be the white matter, and then the darker gray parts here would be the gray matter. So the gray matter kind of covers the white matter. So our desire to link structure and function in the brain is not a new concept. You may have heard of the term phrenology. How many people have heard this term? Okay, a decent amount of you, which has been, sorry to say, debunked at this point. But it's the study of like bumps and the shape of the skull with the aim of linking those bumps and things to different personality traits. And as far as I know, it's no longer practiced today. But funnily enough, I did come across this more modern image, which you can buy on Amazon if you would like. But it's called the Phrenology of Trump. And instead of using the skull structure to determine personality traits, they use the structure of his hair. So they're probably equally scientifically sound. So this whole idea of phrenology, it sounds kind of ridiculous now, but the general notion of certain parts of the brain being specialized in some way is a belief that still holds true in many ways. Of course, we acknowledge that it's more complicated than that. We do still believe that certain regions have some general specializations, and this idea is known as modularity. But we also believe that these regions are interconnected and are part of larger, more complex networks kind of all over the brain that we can study using things like neuroimaging. And of course, some people could argue that neuroimaging is kind of a more sophisticated type of phrenology, and perhaps in some ways it is, but you know, we do the best with the technology that's available to us. And it's still better than that, I would say. So not long after this phrenology craze, attempts to separate out the parts of the brain based on differences in the surface architecture were made by a gentleman named Brodmann. Brodmann very meticulously looked at the brain areas of humans and other animals through a microscope after they were dead, to see how the structures, or the cytoarchitecture, a term you know well now, in each region differed. And through your psych courses, you may have heard of Brodmann areas. I know it's kind of a dying thing. People don't really talk about these areas as much as they used to, but you might still come across it. For example, Brodmann areas 41 and 42 over here are considered the auditory areas of the brain. And then we've got kind of in yellow here, this Broca's area, which is Brodmann areas 44 and 45. So that's more related to speech production. And then down here, this darker orange bit, That's the Wernicke's area, which is more involved in language comprehension. And this, at the time, was a big deal. It certainly still has some relevance today, but thanks to neuroimaging, we can be even more precise now in determining how each brain region functions. And I should also point out, an important thing is that the architecture of the brain, the structure here, and the function of these regions are not always perfectly aligned. But this can at least give us a general idea of where these regions are located. Like he was doing a pretty good job of identifying the auditory cortex here, visual regions here, et cetera, et cetera. So something that's unique about the auditory system compared to the visual system is how much processing occurs before sound even gets up to the cortex. In vision, the primary visual area, called V1, in the cortex is considered low-level processing still, because not a lot of processing occurs before that point. On the other hand, the primary auditory areas are considered to be fairly complex processing up in the cortex. As you've already learned in this class, processing already occurs in the ear. We know about the cochlea and how we get these complex waveforms that enter our ear that have a lot of different frequencies in them. And then in the cochlea, that sound is kind of separated into its frequency components. So this is the tonotopic mapping. So that's already some processing that's happening before we get up to the cortex. And that tonotopic mapping travels and is processed here in the kind of brainstem regions and up here, and then finally it reaches the auditory cortex. So we have a lot of kind of way stations where sound is being processed before we even get up to the brain. And a reason for this may be that all of this auditory information is coming at us very quickly, very complex waveforms. I'm talking pretty quickly right now, and you need to parse what I'm saying, while also ignoring kind of ambient noise in the hallway and other distractors. We have a lot that we're trying to accomplish in a short amount of time. So the more rapid processing we can do in these areas before we get up to the brain, up to the cortex, where processing happens a bit more slowly, the better our chances of effectively processing all of the auditory information around us. And one thing you'll notice here is that the pathways that are being shown here, we're starting on one side of the body, so we're looking at the ear on this side of the body, but we're showing the processing happening on the opposite side of the brain, and this is known as contralateral processing. But what should be pointed out is that we do have both ipsilateral and contralateral processing. So sound that reaches one ear goes to both parts of the brain. So don't be confused by this diagram. It's true that the contralateral processing is stronger, so information that reaches my right ear is processed more strongly in the left hemisphere, but it does get to both parts of the brain. So let's look more closely at the areas in the auditory cortex on this lovely human brain. So here's the brain from the side. So say you're looking at the side of my head without a skull and whatnot. And here is the Sylvian fissure, this line right here, which is the biggest fold in the brain. So if I were to draw a cartoon of the brain, like so, it would look something like this. So this is the fold. And here's the Heschl's sulcus, which is roughly where auditory regions can be found. And if we slice this brain open, so let's just take a big knife and just cut it like so, that's where we can look inside the brain at these auditory regions. So the tricky thing is that a lot of auditory processing happens within this fold, so it's harder to see from the outside of the brain, so we need to look inside that fold to find those auditory regions. So once we get inside, we see the Heschel's gyrus here on both sides of the brain, and that is generally considered to be the location where primary auditory cortex is housed, so kind of the initial stage of cortical processing happening here. So if I were to draw out the primary auditory regions, I might do something like that. And in the more frontal part, in front of the Heschel's gyrus, we've got the planum polare on both sides, and then behind the Heschel's gyrus, I've got the planum temporale, so here and here. And then we've got these sulci here as well. So if I were to draw kind of the non-primary regions where maybe higher level auditory processing occurs, it might look something like that. So taken together, this is kind of generally the auditory processing regions within the temporal lobes. So not unlike our very unique fingerprints and our very unique pinna of our ears, our brains are quite unique too. So focusing on the primary auditory region, the Heschel's gyrus is shown here on these different kind of chunks of brain that have been cut out, but here's the Heschel's gyrus in each one of them. And you'll see that they look kind of different for everyone. The size and the shape differ. And fun fact, you may not know this, and you may have this yourself, but not everyone has just one Heschel's gyrus within a hemisphere. You can actually have 1.5 or 2 within a hemisphere. And it's fairly common, actually. So if you ever get your brain scanned, you can see if you're one of those cool people that has multiple Heschel's gyri in one hemisphere. And we don't know yet if there's any relationship between like musical expertise, like if you would be some sort of musical savant because you have multiple Heschel's gyri in a hemisphere. So we can't necessarily draw those conclusions yet, but interesting nonetheless. So these shaded regions are the primary auditory regions being defined cytoarchitectonically. So the cell structure of these areas is how they're defining these regions. And the way to accurately define the primary auditory regions and auditory regions in general is actually an active topic currently. As I mentioned earlier, the structure and the function of the brain are not always perfectly matched, so I can't just say, okay, here's the Heschel's gyrus, this is exactly where the primary auditory functioning happens, but it's a rough guide at least. So one way we can go about exploring the parts of the brain necessary for certain functions is by studying people with brain trauma or strokes, and finding people who have certain brain areas that no longer work and determining what it is that they are no longer able to do or what deficits that they have as a result. Of course, this is messy and imprecise because, as you probably know, brain damage rarely affects just one very specific region. So it's hard to do controlled studies of damaged brains like this. So an alternative is actually to create the damage ourselves, or get a licensed surgeon to do it, by very carefully removing brain parts via things like a cortectomy, removing parts of the cortex, or a lobectomy, removing an entire lobe. And I acknowledge this is not the most ethical thing to do to people or animals, but if you're doing something to relieve them of epilepsy, for example, this is a good population to study because they're already getting this surgery to help them, so then you can kind of study their functioning afterwards and how things differ. And of course, we're still dealing with the issue of these brains technically not being healthy and normal, so the conclusions that we can draw from these brains and associate them with normal brains is tricky. So how can we determine what these brain regions do? So let's say you've got a person with brain damage. What kind of tests can you do? You can start with something called dissociations. And again, this is possibly something you've heard of in other psychology classes, but if not, I'll go over it. If so, I apologize. You can just zone out for a second. So dissociations, let's say a subject is bad. You give them 2 tasks, and the subject is bad at task A, but they do well at task B. Then we can say, okay, perhaps they're bad at task A because they're missing this brain region, but they're good at task B because this other brain region is intact. Of course, there are problems with making these, drawing these strong conclusions. Like, what if one task was just more difficult than the other? What if them being bad at this task has nothing to do with that brain region at all? So doing a single dissociation like that comes with its issues. But double dissociations are one step better. So take 2 people who have 2 different brain lesions and have them each perform task A and task B. If one subject performs well at task A and poorly at task B, and the other one performs well at task B and poorly at task A, then we can start to narrow down possible functions of these missing brain regions that they have. And such techniques are considered gold standard in cognitive neuropsychology these days. So this is kind of the ideal scenario. All right, so let's look back at some earlier studies looking at speech and music processing, for example. There are some people who have had damage to their left hemisphere around the Broca's and Wernicke's areas that lose much of their speech production and perception. But their ability to recognize music and perhaps perform music may still be okay. On the other hand, damage to the right hemisphere may significantly impair certain aspects of their music comprehension and performance, but maybe their speech is fine. And this is not to say that language and music are processed exclusively in one hemisphere or the other. That would be an inaccurate generalization. But there may be critical aspects of early speech and music processing that take place in certain regions, and when those are damaged, it messes with this entire processing pipeline. Also, there are differences between left and right-handed individuals and other individual differences that exist, and none of these are a guarantee for how a person will process sounds. For example, right-handed people tend to have language processing more in their left hemisphere. And left-handed people tend to be a little bit more bilateral with their language processing. Women apparently have more bilateral processing than men. I don't know why. So again, we're seeing these differences across populations and across individuals in terms of how things are processed. So it's difficult to draw really strong conclusions like, okay, the right hemisphere is clearly the music hemisphere, left hemisphere clearly language. And popular science likes to make claims about hemispheric dominance and lateralization, referring to people as being right-brained or left-brained. I think people who are right-brained are supposed to be more, is the left brain more analytical, right brain more holistic and artistic? So you've probably seen these quizzes online. Go ahead and take them if you want for fun, but proceed with caution when you read into these things. Most of them aren't very scientifically valid. But there have been some interesting studies on right versus left ear advantage in perception. For example, if you play speech or melody to both ears, people will have a tendency to listen more with one ear than the other. So it tends to be a right ear advantage for speech, which would align with our strong contralateral processing, where we'd be processing language in the left hemisphere. And the same thing with left hemisphere advantage for music processing. And that's what Kimura found, this left ear advantage in melody perception. And you can put a little asterisk next to that one. You might get a quiz question about that. So again, left ear advantage. If we're playing melodies in the left ear, they'll be processed in the right hemisphere. So this is this contralateral connections idea. But there was also an interesting follow-up study by Bever and Chiarello, which found that actually for musicians, this lateralization was reversed, suggesting that perhaps the way musicians process music is more akin to how non-musicians process language, so processing music more analytically. So in that sense, we can see more of a connection between music and language. And the idea would be that musical processing with more experience kind of migrates over to the other hemisphere. So the more language dominant side of the brain. And that all sounds really profound and interesting. But follow-up studies, unfortunately, haven't been quite as convincing. And this right-left hemisphere distinction between where things are processed seems a lot less clear-cut than was initially believed, and outcomes have been pretty mixed. And in my experience, and also from what I've read from other studies, processing of sounds seems to be more bilateral than people previously thought. However, it may still be the case that there are differences in the types of processing that occur in each hemisphere. For example, it may be that rapid timing information is better in the left hemisphere, so being more sensitive to rhythm and rapid speech processing, which would align with the whole language processing on the left hemisphere. And the right hemisphere may be more sensitive to melody processing, which tends to change more gradually than speech. And also it may be good for processing spectral changes, so changes in the frequency content of the sounds as opposed to the timing information. So there is some evidence to support that. All right, so Isabelle Peretz, a name you will probably come across in this class, a scientist in Montreal, and one of the big names in the study of neural and cognitive aspects of music perception, has looked at several patients with neurological deficits specific to music. For example, this patient IR had severe damage to both temporal lobes and the right frontal lobe. And despite all of this damage, the patient's verbal abilities were still intact. But what was impaired was their ability to recognize melodies. So even very basic ones that they knew from childhood, they couldn't recognize them anymore. And also basic pitch discrimination, so the ability to determine just distinguishing differences between notes. That was gone too. What was interesting, though, is that the patient could still perceive the emotional content of the music, so they could tell whether the music was happy or sad, even if they couldn't recognize the melody or distinguish the pitches of the tones, which is kind of interesting, but it may be that that kind of frontal region that was left over was doing some of this emotional processing of the music, or some other part of the brain that was still there. And damage to the temporal lobes can affect many different types of processing, pitch contour or pitch interval perception. You can get tune agnosia, which is, again, the inability to name familiar tunes. And there are also cases where left hemisphere damage can affect speech processing, which would make sense if we process language in the left hemisphere, while seemingly leaving music processing untouched. And an example of this, actually pretty profound example, is composer Shebelin. He's a Russian composer who lived from 1902 to 1963. He had a couple of strokes, 2 or 3 really major strokes that resulted in his language capabilities becoming impaired. He just couldn't speak anymore. But his ability to compose music remained intact right up until his death. And in fact, he composed a symphony on the year of his death, 1963. I think it's called Symphony Number 5. So if you're interested, if Russian composers from the middle 1900s are your jam, you can check out just how good he was at still composing music, even though he had these strokes and had massive brain damage. So that's pretty fascinating. And another interesting thing about music is that patients with Alzheimer's disease, as I think you saw on Monday in the documentary, whose memories have really gone, they often still have a very good memory for music, especially music that they grew up with. I think that's a pretty moving documentary. Did you guys like it? Did anyone get a little teary-eyed? I totally did. I've seen it like 3 times, and my eyes water every time. Get really emotional. And my grandma, who passed away a couple of years ago, when I would visit her, I would bring like an iPod shuffle with her favorite music. And she had major Alzheimer issues, problems with memory, but when she would hear music that she recognized, she just would light up, and it was pretty exciting. So if that documentary didn't convince you, I also give a ringing endorsement to giving music to your elderly relatives. All right. So far we've focused on lesion-based studies, or studies where we look at damaged brains to try to determine the function of each brain region. Of course, these studies do come with some criticisms. One being, how much automotive engineering can you learn by smashing parts of a car engine, which is effectively what we're doing. And it's true, there's a limit to how much we can learn with this approach of kind of damaging all these different brain regions and seeing how that affects brain function in general. Because as we know, lesion brains are not normal, so how can we presume to know how a normal brain functions when only looking at abnormal brains? And we know, or you will learn to know, that the brain is very plastic, meaning that things change with experience and with damage. So the brain processing in lesion patients may have been altered in a way that is very unique to their particular situation. And with respect to plasticity, for example, we can take people who are blind, and studies have been done by people in this department, in fact, that have shown that people who are blind, their visual cortex, since it's not being used for visual information, has a tendency to kind of being taken over by other processing, by other senses. For example, touch, which is very important for reading Braille, can show up in the occipital lobe in people who are blind, and possibly even sound. As we've seen people who are blind that seem to have this super sensitive hearing, they can do things like echolocate, where they make like clicking sounds, like, and they hear the sound kind of bouncing off of different surfaces around them, and they can kind of navigate through the world that way. And there's actually a podcast. Has anyone heard of the podcast called Invisibilia? I see one nod. Yes. It's an excellent podcast, in my opinion. They have really interesting episodes, but one of the episodes is called Batman, and it kind of looks at people who do this echolocation and kind of understanding their world, but it also kind of addresses the idea of how sighted people can put limitations on what they expect from blind people and how they can, what they can accomplish in the world, and how those expectations can actually really set back the capabilities of someone who would otherwise excel. So yeah, it's an interesting episode. I recommend it. Not required, but if you like podcasts like I do, definitely give it a listen. Okay, so in summation, it's hard to draw strong conclusions about how a normal brain works by only studying abnormal brains. So we need a way to study normal brains as well. And as I mentioned before, effects of lesions can be difficult to localize precisely. So what is a poor scientist to do? And that is where we get the neuroimaging of normal brains. Yay! So there's been an explosion in neuroimaging research to answer all sorts of questions, not just, music or auditory related, but many other domains as well. And various techniques are available to us, and there are trade-offs for each one of them. Some have better temporal or timing information, temporal resolution. Others have better spatial resolution. And some involve very noisy machines. Others are very quiet. But each one of these techniques is getting better all the time, and we're making rapid progress in improving these technologies and analyzing the data that we get from these devices. And it's important to point out that the best technique will depend on your particular research question. There's no singular approach that's perfect for all research questions. And when possible, it is a good idea to combine multiple approaches to answer these scientific questions. So not sticking to just one technique, but seeing if your results using one technique align with your results using a different technique. All right, so some quick neuroimaging related terminology. As we know, brains are three-dimensional objects, but pictures of brains are two-dimensional. So we need ways of viewing these three-dimensional objects in a two-dimensional space, and ways of verbally describing these locations as well. So that's where these terms coronal, axial, and sagittal view come in. So coronal view so these are different slice orientations. And the coronal view, think of as, you got my profile right here, slicing, starting with my face, you're slicing my face off, and then continuing back. Okay, so these are coronal slices. You can think of it as like a crown, like a headband. It's a kind of good way of remembering it. And then we've got the axial view. I have really crude ways of remembering these things. Bear with me. I think I've seen too many Game of Thrones episodes. So axial, you can think of as like a beheading. So if you chop off someone's neck and work your way up, these are the axial slices. Sounds upsetting, but you'll probably remember that because of that. And then there's sagittal. I don't have a good way of remembering that one. If anyone has yes?
Sagittal actually means archer in Greek, I think, and like from a view from an archer, it's from the side.
Ah, you're good. That's okay. So yeah, use that. So think of archery from the side. So these slices are basically like slicing this way. So you get slices where you're looking at the side. Nice, thank you. I will use that in the future. Okay, so and what else we got? So we've got dorsal being the upper part of the brain, like think of a dorsal fin on a dolphin, like so. And then we've got ventral, which means belly, so looking at the kind of bottom part of the brain. And then we've got anterior, meaning the front, also known as the rostral part. And then posterior, the back part of the brain, also known as caudal. And then you'll hear terms like lateral, which means kind of the sides of the brain. And then medial, going more towards the middle of the brain. And just to confuse you, just for funsies, the convention tends to be that the right hemisphere is located on the left side of the image, and the left hemisphere is located on the right side of the image. And this goes back to old clinical practices, but oftentimes is still used today. So just when you're reading research papers, be sure to check to see how they're labeled, because right might not mean right. Right might mean left. Yes?
So is it coming from the front, but it's like reversed?
Yes. So I think the idea was that doctors used to kind of, when looking at their patient, is like the doctor's right side would be the patient's left side. So they would kind of flip things when they're describing it. Something along those lines. But yeah, indeed. So let's look at some of these techniques, starting with PET, or positron emission tomography. So here we're measuring blood flow in the brain by injecting a weak radioactive tracer into the subject, which goes into their bloodstream. So technically this is an invasive technique because you're introducing something into the subject's body. And the reactions that take place once the tracer is injected are pretty complicated, but the tracer travels up to the brain and resides there for a period of time. And it shows up more strongly in regions where more blood is going. So it targets specific brain regions. And it has decent spatial resolution. I think depending on what tracer you use, you can get different resolutions, but on the order of several millimeters to maybe up to a centimeter. Another nice thing is that it's quiet, which is good for auditory experiments. And it can be combined with MR or CT, so you can get like a nice anatomical image and then overlay the kind of activation maps that you get from the PET scanning onto those to get a good idea of where they are in the brain. Disadvantage, the temporal resolution. It's slow to respond. So first, you inject your subjects with this tracer, and then you have to wait like 45 minutes for the tracer to make its way up to the brain, and then you can scan the person's brain while presenting stimuli, and then you have to wait some more time to get a response, on the order of like tens of seconds to get a response to that stimulus. And one obvious problem with that is that sound changes very rapidly over time. So this technique is not great for discriminating very fine temporal differences between sounds. And it averages activity over a long time period. So basically you can't use this for very fast timing comparisons between sounds. And there's a limit to how long the study can last, because the tracer will eventually decay. And another disadvantage, just a tiny thing, is that people aren't always keen on being injected with radioactive materials. So it can be hard to convince people to actually participate in your study. On to MRI, or fMRI. Functional magnetic resonance imaging is what it stands for. And this is a technique that's rapidly increased in popularity in recent years for the neuroimaging of humans. And what this technique does is it measures blood oxygenation, which again can tell you something about how active certain brain regions are during certain tasks. One awesome thing about it is that it's totally non-invasive, so you don't have to inject people with anything, you don't have to cut them open and remove things. They can just lay in a scanner and you can measure brain activation. And it's got very good spatial resolution. So at a 3 Tesla scanner, for example, which is a moderately strength scanner, moderately strong, we can get down to 1 to 2 millimeter resolution of voxels. And a voxel being basically a 3-dimensional pixel. So think of it as a little cube. That's how small we can get in terms of distinguishing different brain areas. And at a 7 Tesla scanner, we can get down to less than a millimeter, so super tiny, which is pretty cool. And at the CMRR here on campus, they recently installed a 10.5 Tesla scanner, which is the strongest human magnet in the world. Pretty cool. Tell your friends. And very recent news, on December 14th, 2017, history was made. The first official image of a human was acquired at the 10.5T, so it was a very exciting day. They had this thing installed several years ago, and they've had to ramp up the magnet and do tons of testing and tons of IRB approval. Craziness. But they finally are starting to run human subjects on actual experiments now, so it's an exciting time to be alive. And this guy here is Camille Urbil, and he is kind of the big man on campus. He's the head of the CMRR, and it's largely thanks to him that the CMRR keeps pushing the boundaries of neuroimaging research. We're at the cutting edge of neuroimaging technology here, which is pretty exciting. For example, we were the first facility to have a 7 Tesla scanner, so we have the oldest 7 Tesla in the world. It's over 20 years old, and it's behaving like it. It's been breaking down a lot lately. But yeah, so if you're interested in neuroimaging research, this is a really good place to be. It's one of the best facilities in the world. And as much as I like MRI and fMRI, I must admit it's not perfect. A major disadvantage is how noisy the scanners are. And this interferes with brain activity in response to the auditory stimuli that we are presenting. So say you're trying to play your participants this beautiful piece of orchestral music, and in the background you've got this going on. That's basically what the magnet sounds like. So we need to come up with ways of working around this, and I'll discuss those in more detail in a bit. But I'm hoping that this will improve over time as well. It's certainly something that is being actively studied, but it is a very difficult issue. And unfortunately, the stronger the magnet, so starting at 1.5, then 3, then 7, then 10.5, the louder these pulse sequences become. So it's an ongoing issue. Oh, and another downside is the temporal resolution is not great. It's on the order of seconds. So after a sound is played, it takes some time for the bold response to kick in and reach its peak activation level. And that's just the physiology of our bodies basically playing against us. So it's hard to really improve that part of it. It's better than PET, but still not perfect. So here's a general overview of this hemodynamic or bold response that we get with fMRI. So on the x-axis, we have time. On the y-axis, we have this bold response that we're measuring. So the first rise, let's say this box here is your stimulus. The first rise can be thought of as the onset of the processing of the stimulus. We get a peak in the bold response around 4 to 5 seconds after the stimulus is turned on, and then after the stimulus is turned off, it takes a little while, several more seconds, to return down to this baseline activation. And this timing differs for different brain regions, different people, different times of day. Caffeine consumption can affect this. I think it actually leads to a stronger response if you consume more caffeine. So, don't hesitate to do so if you participate in a study. Fatigue can have an effect, alcohol, various medications. So these are just kind of general time estimates. So what is it that fMRI is actually measuring? In broad terms, it's an indirect measure of neural activity. So say we have a change in brain activity in response to a stimulus. That leads to a metabolic response in our bodies, which leads to a change in the blood flow and a change in the blood oxygenation, the amount of oxygen in the blood, and ultimately a change in the image signal strength, or what we think of as the fMRI activation. So we often make the mistake of saying we're measuring neural response to the stimuli, when in actuality it's a measure of this signal strength that we get as a result of all of these things, which we ultimately think are due to the neural response in the brain, but this relationship is not a direct relationship. But based on simultaneous fMRI and electrophysiological recordings, it does seem to be pretty likely that the bold response does actually reflect this neural response. So that's promising. Indirect, but probably accurate. Now, another point of caution, using fMRI puts some limits on the type of questions and the type of experimental design you use. fMRI can only measure differences between conditions. So the idea is to see which brain regions are more active for one stimulus compared to another. So let's say I want to see which brain regions are active in melody perception. In order to do that, I need something else to compare to. So I'd have a condition where I'm playing sequences of pitches, which is creating a melody, and that will give me some sort of activation in the brain. But in order to determine if that area is specific to melody perception, I need to compare it to something else, like a sequence of sounds that varies in some other way, like varying in the timbre of the sounds, like playing musical instruments, but they're all playing the same pitch. So I get a sequence of sounds where the pitch or the melody stays the same, but the timbre of the notes is the thing that's changing, and I compare that to pitches that are changing, but the timbre remains the same, for example. So I can compare these conditions and see which regions are more active to variations in pitch and which regions are more active to variations in timbre. And you can also compare pitch, for example, to no pitch sounds, like noise, for example, and compare those. And these comparisons can help you zero in on regions most sensitive to pitch or melody. But it is a very tricky question to answer, and it's actually something scientists are still working on today, myself included. Like, is there a true pitch center of the brain? Jury's still out on that one. We're not sure. We're gathering evidence. It may be that there isn't a region that responds exclusively to pitch sounds, or perhaps the resolution of the scanners we're using isn't good enough to distinguish it, or perhaps there's something about pitch that's being processed in these areas. So it's kind of a complicated question. And then we could, for example, use a vision study where we want to find a brain region that's sensitive to faces. We would want to compare a bunch of faces to some stimuli that don't have faces, like places or houses. And that can help you zero in on what region of the brain likes faces the most. So here's an example of the type of images you'll see in these fMRI studies. Here we show both coronal and axial slices. So you play stimuli to your participants, and here we had baseline conditions where no stimuli were playing. That's these gray bits here. And then we compare that to when there's pitch varying or when timbre is varying. And it's kind of a block design, so think of this as like 1 5-minute run, and then you would have several runs as you're running this experiment to gather more data, and then kind of average it together to see which regions are responding to pitch and which regions are responding to timbre. And we were getting nice, strong activation in the auditory cortices for both of these, which means we're on the right track at least. Here's another example of fMRI figures you may run into. Instead of looking at these flat slices at different orientations, like in the previous slide, you can also show activation on inflated brains, like this. So we take all of these slices at different orientations, we basically reconstruct a 3-dimensional view of the brain, and that's where we get this inflated brain. And it not only looks cool, in my opinion, but it can make it easier to see most or all of the brain in one image as opposed to kind of flipping through multiple slices. And it can help you orient yourself, like, here's the temporal lobe, no problem. And I have an easier time finding auditory regions, for example. Remember I said that they're hidden inside the Sylvian fissure, which is this folded up tight region where you have to cut off the brain to look inside to see those areas. Or with this reconstructed brain, I can basically treat it like a balloon, blow it up a little bit, inflate these areas, and then I can see inside these folds. So like these light areas are the sulci, or the gyri, and the dark regions are the sulci, and then within this fold I can see inside there as well. So we're basically pushing out those folds so we can see what's inside them. So let's get back to this issue of MRI scanners being noisy. One way to deal with it is through sparse temporal acquisition. So instead of having the scanner constantly making noise, which is the result of constantly collecting data, a standard practice for non-auditory experiments, what we would do is we add several seconds of silence in between data acquisition so participants can hear the stimuli without it being contaminated by the scanner noise. So instead of this like, we do something where it's like, silent period, silent period. And within those silent gaps, we play the auditory stimuli so that participants can hear it with the least amount of acoustic contamination. And that makes a difference for the bold response that we're measuring. So if you've got this noise on in the background all the time, and on top of that you're presenting these stimuli, your bold response is going to kind of stay elevated because it's hearing this noise the whole time. Whereas if you have these blips where you're acquiring images, then a silent gap where you're playing your stimuli, the bold response can respond to that stimulus, and then it can have time to drop back down to baseline before going back up again. And that helps us make better contrast, so we can make comparisons that are a little bit easier, sound versus baseline, compared to this situation. So there are some other ways to use MRI, ways of looking at brain structure and function using this MRI equipment that we haven't discussed. One study you'll explore later looks at something called voxel-based morphometry, which is a way of comparing, for example, gray matter and white matter and comparing the thickness of these tissue types across the brain. And this information can be combined with fMRI. So think of MRI as your structural image, looking at the anatomy of the brain, and fMRI being the function within those brain regions. And then we could see, like, okay, the auditory cortex in this person has thicker gray matter, and they also are really good at playing music. So there's a correlation we can make as far as the thickness of the brain in that area and, you know, their skill level, for example. Another one is DTI, or diffusion tensor imaging. And this, you're looking at connectivity within different brain regions. So kind of looking at these tracks to see which brain regions are connected to which. It basically measures the direction of water diffusion, and it looks at axon bundles and their myelin. Lots of fancy stuff going on. But the best part is that it makes these really cool looking images. So the different colors are related to the different directions. And we have resting state fMRI, which is a very large field of research. Tons of people do it now. And what you do is you get a subject to lie in the scanner and do nothing. So in that sense, really easy experimental design. And then you look at the functioning of their brain regions when they're in this resting state. So here you're looking at which brain regions are correlated with one another. So you notice that, brain activity is going up in this region over here, and it's also going up in this region over here at the same time. So you can kind of correlate brain activity even in this resting state. Because it may shock you, but our brains are still active when we're doing quote-unquote nothing. Okay, so moving on to other neuroimaging techniques, we have EEG and MEG, electroencephalography and magnetoencephalography. And these look at the electrical and magnetic fields generated within the brain. And so both of these are generated by these very small electrical currents flowing in the brain, and we're measuring them from outside the brain, so we get these tiny little fields of energy that we're measuring. So an important part of these measures is successfully filtering out kind of non-brain related electrical currents, and then filtering further to only focus on the electric currents that are specific to your task that your subjects are doing. So it's got really good temporal resolution. The timing is great, very precise, on the order of milliseconds. And it's quiet, which is very suitable for auditory experiments. So we don't have that loud machine, like both of these are quiet. This is EEG up here, this is MEG. EEG is super cheap. You just have to pay for this like electrode cap, which is much cheaper than getting an entire MRI machine or an MEG machine, which is really expensive and is basically like a really sophisticated electrode cap. So there's some downsides for sure. We have to sacrifice spatial resolution here. It's really bad spatial resolution. We don't know exactly which brain regions are responsible for which like little waveforms that we are measuring. Definitely far worse than fMRI. Also, it's really tricky to kind of remove artifacts with this. So getting kind of the non-brain related activity out of the signal that you're measuring, which I'll talk about in the next slide. Okay, so here we're looking at EEG, and this is measuring electrical fields based on millions of neurons in the brain. So like fMRI, with each electrode, with fMRI, we're looking at the voxel level. With EEG, we're looking at the electrode level. We're looking at population responses. So not a single neuron. These are not single neuron measurements. They are population responses. So we're measuring a wave in response to the stimulus. And so we have to do lots of measurements and do lots of averaging to get like a nice clean signal out of this. And we have to remove artifacts. So muscle activity, eye blinks, people blink, that's just what they do. And that causes huge artifacts in these signals that you're measuring. So you have to extract those artifacts. If someone has a cell phone in their pocket, that can cause electrical signals that are not the signal you want to be measuring. So make sure people don't have cell phones in their pockets. Chewing gum basically ruins your experiment. Do not let your subject chew any gum. That's huge muscle activation that would ruin your signal. The humidity of the air. If the air is too dry, that can interfere with your signal. And Faraday cages are something that people use to basically block out electrical signals from surrounding areas. So if you have like power lines overhead of the building that you're in, it's not going to interfere with your signal. So these are very tiny little signals that you're trying to measure. And then you've got this electrode cap. So here we're looking at like an aerial view. Here's the nose of the person, ear, and ear. And then each of these is another electrode. So F is for frontal, T is for the temporal ones, O is for occipital, C is for central. Odd numbers are on the left side, even numbers are on the right side. So you can get a general orientation of which electrode is located where. And here's an example of an average waveform based on lots and lots of measures, all averaged together. The signal that you get directly out, the raw signal, looks way worse than this. And the fancy term for this is an evoked potential, or in this case, an auditory event-related potential. And it's a wave evoked by a certain sound. So each of these peaks and dips is called something specific. The first little ones here, labeled with Roman numerals, are believed to come from very early stages of processing down in the brainstem, and they're fairly automatic responses. So whether or not a person is paying attention, you will get these responses. Then we've got kind of middle latency responses here, and then later waves here, which are thought to come from higher level processing. So attention, they're responding to a task, for example. That's where we see differences in these later waves here. So this might also be a quiz question. Put a little asterisk next to it if you're asked about which waves might be involved in attention, for example. Maybe something around here would be a good guess. And so you see that we have these N's up here, which stands for negative, and P down here, which stands for positive. Again, this has to do with old conventions. Way back in the day when they used to make these recordings, apparently when it would come out on the sheet of paper that was doing the measurements, recording the measurements, the negatives would be up and the positives would be down, and that convention has stuck for a long time, but people are starting to flip it. So be sure to look at that in the research papers that you read to see if the negatives are actually still up or if they've flipped it to be more sensible and have positive peaks on top. And these numbers here are related to the timing after the stimulus onset. So P3 here, also known as P300, means the peak 300 milliseconds after the stimulus onset. P2 would be 200 milliseconds after. P1, 100 milliseconds after. Same thing up top here. All right, so we discussed this already. Automatic responses found in the auditory brainstem response are these early latency waves. And then the later waves are involved in attention and task performance responses, so kind of higher level processing more in the cortex area. One thing we haven't discussed is mismatch negativity. So this would be a response to an oddball stimulus in an otherwise regular sequence. So let's say I'm playing a bunch of, tones to you that are the same pitch, for example, boop, 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 and then suddenly I just go boop, and have one that is a different pitch. That could generate this oddball response in your waveform at around 150 milliseconds. So a blip in your waveform, a mismatch negativity. And similarly, there's something called early right anterior negativity, or ERAN, which is at around 200 milliseconds. And this is a response to unexpected musical syntax. So say you're listening to a piece of music, all is going well, suddenly you hear a note that's like out of tune, for example, and that defies your expectations about where the musical structure is going. That can generate this sort of response, an unexpected sound within the musical structure. Okay, any questions about anything? All of that? I know it's a lot to go over. Neuroimaging is really complicated, and going over many different techniques within 75 minutes is a feat. But hopefully you kind of got a general overview. All right, with that in mind, read Chapter 4 if you haven't done so already. Next week, read Chapter 6. Chapter 7 is optional. I think you already read Chapter 5. If not, go ahead and read that too. And then there's background reading on Moodle related to neuroimaging if you want to check that out. And don't forget to take the quiz for this week.